Okay, so welcome back. Thanks for coming in again.
Thank you.
Obviously, the first time wasn't too bad.
No, I don't know the result yet. I haven't seen it.
So what are we going to talk about today? We're sort of taking a similar vein to last time. We're going to talk a little bit more around the history of science and thinking.
No, it's some history involved, but I use the history to illustrate a theme or an obsession I have, which is the necessity for doing interdisciplinary work. And I'll try to explain too. Well, let me just start with an example. I'm going to talk about COVID, the thing of the moment with the bug. So, and I want to relate that to earlier science, and I think you'll see why. So, here's my first slide about science and its connection to magic and religion. And there's something that gets up my nose a lot where people use the idea of evidence-based science. Well, I don't know what other science you would have, non-evidence-based science, which is whatever.
I can show you some, but maybe we'll do that another time.
Do that. But, let me start with this to set things. In 1348, it's a very famous time in Europe, there was a plague, the Great Plague, and 1/3 of people died in France. And so, of course, all England rejoiced. But then they got hit with the plague too. And so the king was a guy called King Louis IX, and he was a very religious guy. And he was very curious too. So he wanted to find out why the plague. So he went to the best possible academics in the world at that time, and they were professors at the University of Paris, at the best medical school in the world. And so they did 2 years of research, and they came up with a solution to this problem. And they proved that the plague was due to a conjunction of Mars, Jupiter and Saturn. That is, all the planets line up in a line, you see them in the sky.
So is that where that concept of, all the planets aligned, is that where that
came from? I suppose so, probably. So the king was very pleased.
We had an answer.
This was a superb grant because, you see, you couldn't prove it was right or not right until the next conjunction. And the next conjunction was 700 years later, and about 2080. Well, it happens, I was in Florence at the time, and coming back with my friend, we actually saw that conjunction coming over the hill, coming down to Florence in the city. We'd been out in Tuscany. So that was very interesting. The king was very pleased. He was the king that built the beautiful chapel near Notre Dame called Sainte Chapelle. It's a gorgeous, beautiful chapel. Anyway, so there we were. So my point I want to make is that we can look at that and we think, how ridiculous. You guys are pretty stupid. Well, are we stupid or not? So COVID-19's come along, and that should be COVID, not COVID anyway. So everyone's excited about that. We've got this terrible drug. And we have all this research, and I'm working on that with my friend Brandon Ronas in America. And what we're doing is putting together two disciplines. I talked about it a bit last time, physical chemistry or surface chemistry, and biology, physiology. And the point I'm trying to make is that the people in the molecular biology and physiology and so on, they're madly working for a panacea for this problem. They're looking for what's it called?
Like a vaccine.
They're looking for a vaccine. And here's what I want to say, that in that research they focus everything on gene technology because that's what they know. And all of biology is about genes and DNA and RNA and sequencing of genes. And I got this virus coming in, and they've got no awareness of the main thing. The virus comes in from the air in a droplet. Mm-hmm. Where's it come from? It comes into the lungs. It comes in and goes into the lungs and goes into the body and does its work, and then it comes out. It comes out. How does it get out? And then it comes out through the lungs.
Through the lungs, yep.
Along with carbon dioxide.
So from the air, through the lungs.
Air and nitrogen coming in.
Yep.
And we've got carbon dioxide.
Well, am I right in that the idea is these are normally attached to water droplets, right?
They're into water droplets.
Yeah, so tiny water droplets in the air, we're breathing them in. Mm. That's then they're then sort of penetrating the lung barrier.
They're getting
into the blood,
well,
and then into your body.
That's right. So they're tackling this vaccine by looking at DNA. Well, they have no idea how the virus gets in.
But am I right in saying so a virus, and the way I learnt viruses when I did biology is a virus is essentially like a small piece of RNA or DNA material encased in. Yeah,
it's encased in a bunch of little balls of proteins.
Yep.
These balls pack around and they pack into a geometric shape like an icosahedron or whatever, and the RNA is inside there. Okay,
yep.
So when they get inside a cell, the changing conditions inside the cell allow the protein coat to dissolve, and the RNA gets in that new environment, and it proceeds to hoist onto the DNA inside the cell and get it to work to make more copies of itself.
Yeah, so essentially because that's what RNA does. It's a replicator, right? So it gets into your cell. It's got all the conditions now to replicate, and away it goes. Yeah. So that's the logical thing is if you can stop that happening, the replication.
Yeah.
Right. But what you're saying is well before that happens, it's got to go through a whole heap.
It's got to get in there somehow.
There. Yeah.
And it's got to get. And the disease, the most important thing is your lungs collapse and you can't breathe.
Mm.
Right. So what's going on? So evidently there's something in the lungs that they don't even know about. They don't know the structure of the lungs. It's a surfactant. There's a lining in the lungs, in the alveoli, the tiny little capillaries, not capillaries, little holes in the lungs, which is a lining, it's called lung surfactant. They're not even aware that lung surfactant exists. And that's clearly important because when the virus comes out, the lungs collapse and the patient dies. He can't breathe. There's something going on in the lungs that stop him from breathing. Because normally when you breathe, for 70 years, you breathe 10 times a minute and you breathe at zero interfacial tension. The lungs expand from collapsed state to about 70 square metres, in and out, 10 times a minute.
And there's no surface tension on
the lung. There's none at all.
During that whole...
well, their model is, they've got a single layer of a lipid soap molecule, which expands or not. But it doesn't work. It's not possible that it works because the surface tension would be far and away too high. You couldn't breathe. Yeah,
because if you've got a limited layer then and you expand the layer, then
well,
how does that... okay.
Anyway, so they're doing all this stuff, but they're not even aware that this lung surfactant exists. So there's lung surfactant lining the alveoli of the lungs, and when the virus gets going, when the virus comes out, the lungs collapse. The patient can't breathe. That's a gross observation. It's called lung surfactant. And there's another layer that I talked about last time called the endothelial surface layer, which is, I'll come to that too, which is a layer on top of all the veins in the body that connect up with the lungs. So let's have a look at the lung surfactant first. Now, the lung surfactant's not the right word, it's a complicated structure. It's the planes there are made up of a special membrane lipid. Membranes in cells, all cell membranes, are made up of a bilayer, or two layers. There's two sides to this thing. The walls are, it's a bunch of compartments. If you imagine that you're doing Japanese origami, you take a single paper and you fold it up, and you fold it and you fold it and you fold it. It's very intricate, okay? And so you get this complicated structure. Now, we've got here a single layer, which is the same kind of structure as a biological cell membrane. It's a very thin layer of what are called lipids. Lipids are double-chained surfactants. They're double chains like two olive oil.
Like a fat. Yeah.
It's a fat, yeah. They're two olive oil chains, hydrocarbons, about 16 to 18 chains of carbon, and they've got a head group. The tails like to be in oil or water. Yep. Oil and water don't mix, and the head groups, they like to be in water. They're hydrophilic. The tails are hydrophobic. Yep. Okay, so the two are confused. They're amphiphilic. The head group likes to be in water. The tails like to be in oil. So the tails get together, put their tails together so they satisfy their oil requirements, and the heads stick out. So you've got what's called a bilayer, two layers of these things, and inside that bilayer, which bounds the cell, there's a myriad other components too. There's proteins and there's this and there's that. And it's a whole forest of stuff going on in two dimensions in this bilayer. And so this bilayer with all the stuff in it bounds the cell. It's like the edge of a balloon. It's very thin, very, very thin, molecular leaflet. Mm-hmm. But without it, the cell wouldn't exist.
It's a thing holding it all together, basically.
Yeah, okay. Yep. But it's a mixture of other things, a whole world there, okay, in two dimensions. Now, the lung surfactant, this lining on top of the alveoli in the lungs, it's a bit different. It's unique in that it has these bilayers, and these bilayers of lipids are planar. There's only one kind there, well, there's two, but there's only, which is very unusual. Usually in the cell, cancer cell or red cell, there's a whole mixture of different ones. Okay,
right.
But it's only got one kind, and it forms a plane, and it's, as I said, it's a single layer, an enormous bilayer, which folds up like origami. And it folds up into this thing. As you breathe in and out, it folds and unfolds. Right? And it does that by, at the corners of these cubes, it forms these cubes or tubes. It's got four little proteins. And these little proteins, called A, B, C, D, they're crucial to put it together. One of them forms a hexamer, like a Lego block. Yep. You've got hexamer, there's 6 arms that join, you can see in the here, it joins the planes together, yep. Sticks into them at the corners of the planes, and then the other ones help it by packing in the right way, so the lipids and the hydrophilic parts, the hydrophobic parts can join, otherwise it would be energetically not very good. So they do that, and so that's the lipid. So they expand, pull right out, you take in oxygen and nitrogen, breathe it in, and then it unfolds and it dumps it into the body, okay? So it does this, as I said, 10 times a minute.
So I'm picturing now your origami things that is I think a really interesting way to think about it. So you have essentially a huge piece of paper condensed down into some sort of structure that now can open up and come back again. And so there's no, going back to your surface tension thing, therefore the surface tension of the paper never really changes because it's just...
No, yeah, that's right. It's folding.
Yeah, okay. So. Right, okay.
So this is very unusual because it involves only a single surfactant, but the geometry is called it's called a cubic phase, and that's interesting too because the geometry that I was talking about, it's called a cubic phase because it's got cubic symmetry, the symmetry of a cube. Yep. You can rotate it, but it's not cubic at all. There's many of these geometries in biology. They occur everywhere, and they're what's called non-Euclidean geometries. They're not just the geometries of spheres,
not just standard, yes. Yeah.
They're these complicated bicontinuous structures. And here's another one, which is a random bicontinuous one that's got channels going all the way through it, and it's a bit like our ion exchange resin because you see it's bicontinuous and the ions come into this stuff which is all twisted and polymers twisting around and so on like that. That, in fact, is a white ant's nest. They make that's a real time white ant's nest. It's got all these channels in it, and they use that to cool the place down to get things in and out and so on. Made out of dung.
But those structures are the most efficient way to create sort of channels through a...
Okay. You've got a bicontinuous structure. So with the ants, you've got channels to going in and channels to go out.
Yep.
With in mitochondria and cells that produce all the energy. Yep. They're bicontinuous structures, folded membranes like this. But the ones in the lungs are a bit special because, as I said, they're made out of this single layer of these fats, these lipids, with all these special proteins. So we know that, and we know if you breathe in, it expands and unfolds and delivers the oxygen and nitrogen somehow, okay? So that's one we've got in there.
So is that flipping you're talking about? So as it goes out, it sort of, I'm imagining now my origami thing as well, it folds out this way and now, you know...
It folds out in both directions.
Yeah, so both directions, so this is breathing in, this is breathing out sort of scenario. Yeah, okay, yep, right.
So let's go on now to the other. That's where the virus comes in through the alveoli. Okay.
Yep.
Now I should go back. The dimensions here are about 40 nanometers. That's 400 angstroms, and angstroms about the size of a hydrogen atom. That's about 400 hydrogen atoms in size. So that's 40 nanometers. That's a nanometer
per whole.
A billionth of a metre, all right. Yep. That's the holes. The virus, this particular virus, is odd. It's a very small one, and it's only 120 nanometres in size. It's much smaller than the others, so it's about as big as these. So it's coming in there, it's going in with them, and it's clearly messing with the bilayers because it's got to push them out of the way, and it's getting in somehow through them, all right? So it gets in there somehow, and off it goes. Now, when it comes out, it's got to go out. It goes out through the veins. We talked about that last time. And on top of all the veins, all the capillaries in the body, there's all these veins with branched roots and so on that connect back to the heart, and they connect through that to the arteries. And they've got, again, the lining that underneath them is again a membrane, a lipid membrane, same kind of thing, and it's on top of it, and on top of all cells too, there's something called a glycocalyx. I'll talk about that too. The glycocalyx involves a bunch of polymers that line the cell, inside this interior of the cell, and the glycocalyx is a bunch of polymers, and you've heard the names of heparin sulfate and chondroitin sulfate and hydrolaurin. You can buy them in the chemist, the hydrolaurin for your eyes.
Well, you've heard of them, but maybe not everybody's heard of them.
No, you don't go to the chemist shop.
But you can use them, yeah.
Chondroitin you use for joints. Okay. So anyway, there's a layer there which is quite large. It's 50 nanometres. So the scale is about the same size as our holes in the lung.
Yep.
Okay, that was 40 nanometres. So. And then on top of that, there's this huge layer called the endothelial surface layer. And nobody knew anything about it until we found out about it last year. Nobody knew anything about it. Now, it's completely fundamental. They couldn't find anything there. They tried to look at it with electron microscopy, everything. They couldn't find anything. They could find a little bit, and I'd say it's 10 times as big as this 50 nanometres. It's a micron, which is it's a millionth no, it's 10 to minus 4, 10,000th of a centimetre. Yeah, a millionth of a metre. And this is, it's 50 times as big as that. And there's nothing there.
Yep.
And yet it stops red blood cells from getting in. So the blood cells are going there, the blood flow is, the blood's going along outside it. And any kind of cells that want to get in there, like cancer cells or bacteria or whatever, they've got to make this journey through this layer here, which is pretty big, about the size of a cell. But it keeps out cancer cells, it keeps out T cells that are fighting the immune response and so on. they've got to get through this and they've got to get through the glycocalyx. Well, we found out what it is and its structure too, and it's the layer, the glycocalyx, is a bunch of polymers that they lay down flat and they stretch out into this hairy thing here a little way, but it's mostly still nothing there. And then it turns out that it's really full of carbon dioxide. The carbon dioxide bubbles, nanobubbles, the same size as these lung surfactant holes, right, are formed by metabolism. Mm-hmm. And the body produces carbon dioxide and it's excreted through the glycocalyx, through this polymer thing, and it stays there as a foam and it's supported by these polymers sticking out a bit. And so to imagine it, we've got this picture here of seaweed with bubbles in it. There's a whole bunch of polymers, if you like, the seaweed with the bubbles in it. And here's another picture of the bubbles. They're all coming together. They're all stuck together like this, and the molecular forces that control them and make them stable, it involves physiological saline, salt water, and the thing, and the polymers. Anyway, they're stable and they hold the thing together.
So is that they're nanobubbles because as the cells are respiring and creating carbon dioxide, that carbon dioxide is then going up through the glycocalyx?
Well, yeah, the carbon dioxide gets through the glycocalyx.
Yep. Because of that, you're talking about the holes essentially are so small. The bubbles as they come through are very small bubbles.
Well, the bubbles is like a frit.
Yeah.
So it's a molecular frit.
Yep.
The polymer.
Yep.
It's like an ion exchange resin.
Yeah.
It's a molecular frit, and the bubbles come through, they pop out, and they
but
form
they're tiny
bubbles
because they've come through tiny holes.
Yeah, and they form.
And they stay tiny bubbles because they're coming through into this salty.
Yeah, and they can't expand, partly because the polymer sticks up is setting a limit to their size too. It's forming a template.
Okay. Yep. Okay.
So what happens is now see these massive bubbles. I showed you these bubbles, nanobubbles of carbon dioxide. But what's happening is that it protects it from, they can flow, but you can't squash on them.
Yeah, okay, because there's essentially a gas sort of barrier there.
The barrier, and so when the blood goes past, it pumps past, it wishes them away, clears it out, and then they come in again.
Yep, sort of keeps bubbling
so
through.
this constant dynamic flow. The carbon dioxide bubbles go out, off they go into the veins, and off they go back into the lungs, okay?
Yep.
So you've got this flow of oxygen and nitrogen in and carbon dioxide out. What the biologists think is they've got binding of the oxygen that comes into hemoglobin, which is true. Mm-hmm. They don't know what happened to the nitrogen, and they don't know. They think the carbon dioxide is tied up with hemoglobin somehow, but they have no idea how it works. So there we are. We've got these two systems through which the virus has to go. Comes in through the lungs.
Come in through the lungs.
Goes out through the veins.
Then it's got to go through the glycocalyx and into the cells.
Yeah.
Does its replicating.
Comes out.
Then it's got to come back out again.
And out through the lungs.
And then it's got to come
circulate.
out. Yeah, well, it comes out through the lungs. So but the lungs are collapsing when people are seriously infected.
When they've got the virus, the lungs collapse. You can't breathe. Okay?
Which is why they're coughing and.
So they've got to go into a ventilator to make them breathe.
Okay. Yep.
So. What's going on? Well, what they've found it provides a hint, and they're going blind. Everything goes blind. They're just like the Paris professors. They say it's DNA, something
because they're trying to attack the RNA and stop it replicating.
Yeah, but they're not dealing with the problem at all.
Yeah. So the image I have in my mind as you're talking about this, it's almost like so someone's coming to your house to break in, to steal some stuff and make a mess. And it's like, how they make a mess. So what they're trying to do is stop them making a mess. they've left the door open, there's no security.
Yeah.
You're not worrying about how they got into the building, how they got through your front door.
No.
You're just saying once they get into the kitchen, make it really hard for them to make a mess.
Yeah.
And that'll stop the mess.
Yeah.
And you're saying, well, if you lock the front door, if you lock the door to the building and don't let them in the first place, then problem solved.
Yeah.
Yeah.
Here, because everything is polarised on DNA and RNA, so it must be the answer.
Yes, yeah. Because
But
that's the code. That's the thing that's actually working.
But
yes.
that's not really a direct connection with the virus coming through your lungs and going out through your lungs.
Yes, it's still come in and done all that.
So if you call RNA DNA, it's like saying, astronomy determines the Earth because we've got a revolving sphere, so it has an effect. So it must be due to the planets. So the logic in going for DNA and RNA is not any different to playing with the planets. That's what I'm saying. So you can be as pleased as you like with DNA and RNA, but it's not exactly that much different to attributing everything to the revolution of the planets, right? It's a really indirect connection. So why can it work? Okay, that's what I'll come to now. We've got something that the biologists and the medicos don't have. We know something about how the virus gets in. We know something about how it gets out. So, a bit of a problem. But at least we've focused on the main problem. The main problem is the patient dies through not breathing. So the viruses do something to the lungs as it comes out.
So if you could stop them coming in or you could stop them destroying the lungs, then...
Okay. Now, one other thing we've got is that we found out that carbon dioxide bubbles kill viruses. Mm-hmm. This is something completely different. We're working on that for killing viruses in on the macroscopic scale. So on top of these veins that have got that endothelial surface layer, the layer of foam of carbon dioxide, the virus has got to run the gauntlet of getting through that. So it makes sense because young kids and young people have got a very healthy endothelial surface layer. They're working, they're producing carbon dioxide. Old people don't move much. Their veins are clogged up with low-density lipoproteins from heart disease, from cancer and so on. The blood flow isn't going, the bubbles aren't circulating.
Yep.
So there's not so much carbon dioxide there to kill the viruses. So the viruses are coming out in spades amongst old people and doing their job.
They're still replicating
young people,
fine.
Carbon dioxide kills the virus. That's interesting, okay. Another key. So then they've found out just by chance that they're trying a drug, small molecule, that's used to control malaria. And it's called hydroxychloroquine or something like that, okay. It's a small molecule, looks like a ordinary soap molecule, nothing special. Anyway, it works to kill malaria. With malaria, there's a parasite that gets inside red blood cells. Mosquito puts it in there, and the parasite multiplies inside the red cell, then breaks out and creates havoc, okay. So this drug, they don't know why it works.
But it stops.
It works on malaria, okay. So how does it work? Well, and what's it got to do with COVID? Because they're using it to play with the COVID virus. They're treating patients and sort of mixed results, but it seems to be working.
But it's a parasite, right?
That's a parasite. Yeah,
so it's not a piece of RNA like a virus.
They don't know how it works.
Okay.
It works for malaria. They're trying it out for COVID, for the virus. Seems to work sometimes. Okay. Mixed results. They don't always work. So what's going on? Well, we could ask the Paris professors. I said they might tell you, the best school in the world. So remember that the virus is coming in little droplets of water through the lung surfactant. It's about the size of 3 or 4 of these, and it's doing something. Now
hang on. So the virus is is the size of multiple of these, so
Three
it's got
of
a barging its way through as opposed to getting through the holes.
Yeah, yeah.
Right.
Okay. So remember, when the lung surfactant's working, it's expanding and contracting. It's a conjunction of work by these 4 proteins, A, B, C, that join the edges together, the walls together, and when it opens up, some of the proteins go out. Protein C and D, B go out. A and D are so-called hydrophobic; they like oil. B and C join them up, the others to the planes, the lipid planes, and they're hydrophilic. Well, C and B, as it unfolds, they can go in or out of the bilayer as it's stretched out. The bilayer unravels, and they can join it to be the nuts and bolts that hit it together when you're assembling it, but they can come out and be hanging around when it's stretched right out, but still waiting there to go in and out. Okay.
So they sort of come back in when you...
Yeah, they come back in or go out. Okay. They've got to work together. So that's their role. Now, so something interesting happens because they use the hydroxychloroquinone. And so what's going on? Well, the hydroxychloroquinone is about the same size as the protein C. It's a small molecule.
Okay.
It's about the same
Tiny.
size.
Right.
And a very curious thing is that they use it on infants. When infants, premature babies, they don't have this lung surfactant developed well, and that's when they get the sudden death syndrome. They die. So they have to use pig surfactant or something, but they now use protein C. And they use it because with premature infants, it's the last thing that develops.
Right.
The lung surfactant, because the last thing
the last thing you need is lungs. Yeah.
They don't need lungs when they're inside the mother. They only need lungs when they get out.
Around, yeah.
So it's the last thing that develops. So premature ones are born without these proteins, and the ones that they need is protein C to complete the ensemble. Well, it turns out this hydroxyquinone works
as a protein C
substitute. Protein C as a substitute. You add it to the premature infants, and they can breathe.
Right.
Okay, so that's neat. That's a clue, okay?
Yeah.
Right, a big clue.
Here's a hint.
So
it's not the same, it's a different molecule, but it mimics the behaviour somehow in this
application. There's a
application.
mimic, the hydroquinone evidently mimics the protein C. It can substitute for it, okay? So we know that, anyway, in ordinary people, the protein C is coming in and out. Now, and it can mimic this thing that helps the lungs expand and contract. Well, what's the virus going to do? It's going out, and the observation is you can't breathe. So it's destroying the capacity of the lungs to open and close, which involves all these proteins changing and so on.
Yep.
Well, the virus has got absorption sites on it. It's a spherical thing, there's nothing special about it, but it can absorb things on the surface as it goes through. So it's causing the lungs to collapse. And what it's doing is absorbing the protein C, which stops the lungs from working.
Yep.
So they can't breathe. So it's going through and taking it out. So the lungs can't work. So you've got to put something back in there. The body can't produce enough. It's got to produce enough. So if you put the malaria drug back in, hydroxyquinone, we know it substitutes for protein C with the infant babies, so it can substitute for protein C with the virus clearing off with too much of the protein C. So it can substitute for it, and so you can restore the lung function in the patient.
Right.
So it all fits together, right? It's all surface chemistry.
Okay.
So that's the story.
So can I just to recap, right? So you've got these 4 proteins, which are the Lego block, as you call them, the things that are allowed to come together and expand. Okay, and then it's this protein C. If you take that out of the equation, like in your premature babies, the whole thing doesn't work because you've got a piece.
That's right. You need all 4.
And you're saying that's essentially what the virus is doing. It's coming through. It's knocking out protein C,
carrying it off.
And so if you put in substitute C, like you do with premature babies, And as long as you put that in at enough volume, then you're replacing it fast enough. Yep.
So there's the premature baby thing. There's the question why the drug works, or if drugs works. There's the question of old people, and we know the old people don't have a decent endothelial surface layer, producing carbon dioxide and sweeping it out. And we know that the carbon dioxide kills the virus, so the virus has got a big job to get out in young people. It gets killed off, but it doesn't get killed off in old people. And we know the virus is going to clear out, substitute, take off the protein C, so we can use the drug to replace it. So it all fits together. It's got nothing to do with RNA or DNA, right? It's all surface chemistry. And so my point is that what I'm saying is that this stuff all hangs together. It's starting to make sense. And it's got nothing to do, the way they're working through RNA and DNA, it's so far remote, it's like saying, well, we'll call on astronomy. Well, in some ways, calling on astronomy to explain the plague, it makes sense because the Earth revolves and it's affected in its revolutions by the planets and the sun. So that's the ultimate cause.
Well, it's... okay.
It's the ultimate cause, so it makes sense.
It's a little bit...
DNA is the ultimate cause, so it makes sense. But it's not quite close enough.
It's a little bit better than the planet's scenario because, again, at the end of the day, I'll go back to what I've got in my head is this, literally this, bloke coming and breaking into your house. And if you do stop him messing up the kitchen, and making a mess, well, then you have solved the problem and he can't take that mess into everyone else's kitchen. But I take your point where what you're saying is, okay, well, even if that happens, as long as you can continue to breathe until the body cleans up the mess, then you'll be okay. But more importantly, if you concentrate on stopping them coming in the first place, it really doesn't matter how the virus, replicates, etc. It's never getting in the door, or you're allowing the body's way
the virus is going to get in,
yes.
But we'll kill it off with the carbon dioxide. That's the body's defence.
Yes. Yep.
It's not DNA thing that does it. And going out, we control it. We control it with a drug. That's different to a vaccine.
Yeah,
and
but
I don't think you can make a vaccine to this virus. We've had enough trouble making a vaccine to influenza, which is related to
it. I guess that was going to be my point, right? So part of the problem with the whole DNA, RNA complexity, apart from the fact that there's a relatively simplistic model of this incredibly complex thing, right?
Yeah.
It's the 4 components and so on and so on. So we get this idea that we understand that well enough, and therefore if we keep going down the path of understanding it better and better, and therefore controlling the way DNA replicates or is degraded, etc., right through to aging and disease and all those sort of things. If we can stop the RNA from working, then the virus can't replicate and therefore it'll stop it.
Yeah, that's a vaccine, yeah.
Which is the concept of vaccine. Now, but the problem with that is as soon as there's a change in the RNA, whatever mechanism you're using to stop it replicating doesn't work or isn't as effective or whatever. And so you're sort of constantly chasing your tail on influenza A, B, C, D, E, F, G, and all the different sort of variations or mutations as they're often referred to, as opposed to, I guess, what I'm sort of hearing from you is you're saying the body knows how to do this and can work all this stuff out if you give it the opportunity. If you've got a dodgy door, let's just make the door a bit harder so the virus doesn't get in as frequently and as easily. So the bounces, the CO2, let's make sure there's plenty of that. Let's make sure there's plenty of this, protein C, so that the lungs don't collapse.
Yeah, there
in the
is
first place.
a normal thing, but the virus carries it off. Its surface is favourable for absorption of the protein C, which is loose. It's hanging around. So it can take it off.
Yep.
And the surface is such that you can take it off.
Yep.
So you can substitute it with that drug.
But it's a very different approach. You're saying...
I'm saying that their dogma...
Yes.
Their
dogma is go after the virus, and you're saying support the body.
I'm going after the virus too with a drug. Okay.
Yes.
Well, that's.
But you're not going after. You're not trying to kill the virus with this drug. You're saying the body knows how to kill the virus. We just need to support it long enough for the body to do that in cases where it can't.
Yeah. Well, you can support the poor old people who've got a lot of virus to deal with because their natural defences with the endothelial surface layer, with that foam of carbon dioxide, are not working because it's not working there. So they've got a disadvantage. So you've got to try and keep them alive for long enough. And the way you keep them alive is to stop the virus from running off. There's lots of viruses in old people because they're not killed on the way through. So, so you keep them alive by substituting this drug. So you can invent drugs, other drugs as well. But the idea that you're going to create a vaccine, that implies that you've got to have an antibody to the virus.
Yes.
That there's an antigen on the virus that stimulates something that requires an antibody to control it. That's a dogma. It's a dogma the same as saying that the planets are going to control it. It's got some truth for some things.
Yes,
But these are different. This is a different beast.
Well, that's part of the problem we face because we collapse everything down into the answers of things. And a really good example of this is your salt table of those that prevent bubble coalescence and those that don't, even though they're all salts. But the other thing is, you're learning kind of biology. You're saying, okay, well, the way that the mechanism is, you have these viruses or whatever, and you get an antibody is kind of the way it was described to me is the body makes a key to kind of lock it off, or and then it can then be removed.
That was the original idea of Macfarlane Burnett that started immunology.
Yeah.
But it was a very crude idea. It's a great idea, but it's crude because it had the notion that every antigen in the universe, the body could produce an antibody.
Yes.
Millions and millions and millions.
Yes.
Well, this is absurd. In fact, there's different ways of doing this that involve nanobubbles, actually.
Okay.
So,
of course.
and enzymes, and it's a different way of thinking, which is emerging, okay? The whole thing is changing. So the idea they've got of making a vaccine with a standard way, they have all their equipment and so on, it's probably not going to work. And the reason it's not going to work is because the foundations that lead to this antibody-antigen thing are not right.
Yeah, but you get this sort of confirmation bias, right? So we've definitely cured diseases, cured viruses with vaccines, right? So because there's some, right? So there's this concept of like, vaccines are a good thing, and then you've got this sort of modern, almost sort of like religion of the anti-vaxxers, right? They're saying, vaccines, vaccines don't work, or right through to, they cause autism or whatever, right? So we've got this whole scenario where there's definitely some examples where you can say, if you take this, then you won't get that disease, right? And that seems to be spread now to the idea of, therefore, the answer to all of these is to create a vaccine for them.
Yeah, I know, yeah.
And yet there's plenty of vaccines that definitely don't work, demonstrably don't work.
Well, they're not vaccines.
No. Well, they're called vaccines and they're administered. And so there's this idea that every flu season, you just wait to see what the flus are. You build all these vaccines for the flus. You get them out. You get your 9 shots or whatever, and now you'll be immunised against this year's flu. Now that, if I was to look at this from a business perspective, it's almost like that's perfect. You just wait to see what the influx is this year. We make all the answers to that because you won't have them, and we sell those solutions to whatever this year's new crop of diseases are, right? But the reality is the body clearly has a way of doing this. Now, it doesn't have a way of doing it as efficiently for an 80-year-old as it does for an 8-year-old, from what you're saying. But because some of times it works, we clearly can create vaccines for certain types of scenarios. We've kind of chased this idea that we can always do that.
Yeah, I know. It's saying always you can do it. But there's one other thing I could say, that what's interesting, I think, is that the question is, how does the virus lose its potency? Right, this COVID thing, after a little while, you go through a peak and nothing more, okay? It damps out. In terms of its
spread across human beings,
you have this herd
immunity.
Okay, so why does it lose its potency?
Well, because we all now have those antigens, right?
You're assuming there's antigen. What if there are no antigen? Well, what was happening is something different. Yep. The virus going out is clearing out with protein C. It's coated with protein C.
Because it took it out.
So when it comes out, it's changed its character, its packing and so on.
Yeah.
So when it comes in again, number one, it's not recognised as an antibody or an antigen because it's coated with the same stuff. So it gets through easily without any trouble, okay? And it's taking this protein C with it. So it's changed by picking up the characteristics from the body, from the lung surfactant. So for the next host isn't bothered by it. It doesn't produce any inflammation.
Yes.
Okay.
Yep.
So the antigen thing is different. I mean, the virus is itself weakening itself. It doesn't want to kill off all hosts because it won't exist anymore.
Yes.
So it's got a reason to do it if you give it a thought process.
If you can give it a thought. Yeah.
But
the antigen, viruses survive.
The antigen antibody thing connected to genes is just not necessarily on. It's the same as the enzyme thing where you have a lock and key and everybody's got the lock and key. Doesn't work like that. That ignores molecular forces, which are all operating, in fact. And that was the thing I was going to talk about next. We started with the planets and the plague and our dismissal of the ancients and so on. But the next one is my game. It's about the connections that go all the way back to Babylon and before, with the spreading of oil on water. It's a very interesting story. It's the first scientific paper. Oh. When they discovered, a guy called Layard discovered in the 1850s the biblical city of Nineveh. The French also, that it was a great astonishment. The Bible was real. They found this thing.
Because they found the city in the Bible.
Found the city, yeah. It was all just a claim old. And so in the city of Nineveh, which is the capital of a king called Ashurbanipal, is a fierce Assyrian. They used to have cuneiform tablets. They wrote on clay and with styluses. And so he had they discovered the story of the flood and everything like that in this ruins of the library. A fellow called George Smith took it all back to England, and it's all in a library called the Ashmolean Museum in Oxford. And so they confirmed all the biblical stories and so on, the flood and all the rest of it. But then they discovered too, a colleague of mine, who's dead now, called Tabor. He was in Cambridge, and he discovered in that library a scientific paper, which had originated, was transmitted from Babylon and was in Ashurbanipal's library. And it was a science paper, and it was about the spreading of oil on water. Well, they didn't use it for any particular scientific purpose. There's heaps of oil in Babylon, so it's coming to the surface. So it forms all these different colours and spreading and so on. And so they used it for necromancy, that's communicating with the dead. Yep. It's a bit like the planets thing. And that was the first scientific paper about, because they had all the rules for communicating with the dead, depending on these films. These films have these different films. Well, the films change colour because it's due to interference of light.
Different refraction.
You get the different refraction, so you get the colours of the rainbow, depending on the thickness of the film.
Yep.
But the spreading of the water, of the film on water, the thickness as a function of oil and so on, that's the first application of molecular forces. And all the molecular forces and all the molecular force theory and so on, which underlies all of this stuff we're talking about, it goes back to that. So there's history there, connections.
1600 BC.
BC, and before. And so it takes a while for these things to get sorted out. And I was saying that all of science is the same. We think we know everything at any given time. We think we know about antibodies and antigens and so on. Not so, and evidence-based science isn't quite what we think it is.
Well, I guess it comes down to what is evidence, right? And so,
well, this is evidence.
So the standard of evidence, I suppose, I guess what you're saying is the standard of evidence, the threshold increases over time, or what can be used as evidence. So that was evidence back then, and it was evidence of something very clear because there was a lack of knowledge of what about that wasn't actually evidence. And same with the planets. So the planets were evidence, right? And because they weren't disprovable, therefore that was accepted as evidence, and then right through to your antigens, that someone has an answer to what's happening, and that seems to work, and we have evidence because, we know the vaccine works because we had all these people who had the disease and we gave them the vaccine and they don't have the disease anymore, and we gave the vaccine to these people and they haven't got it. So therefore we have evidence of the fact that even though we don't know why it worked because this happened. And then we have this sort of like, okay, well, there's degrees of it, right? And even I remember when I studied essentially the germ theory of disease, if you like, was we know that this particular pathogen or this particular thing causes the disease if it's in all cases, right, where there's symptoms. It's got to be there in all cases, and it's got to be able to be taken from an infected host and put into an unaffected host, and that host has to develop the symptoms. And then because that doesn't always happen, there was a sort of like a bit of a rewriting, if you like, or at least as I remember it, is saying, okay, well, no, it's a susceptible host. Mm-hmm. So you've got to be able to take it from someone who has the disease and the symptoms, and you've got to be able to place it into a susceptible host, because if you put it into someone who's not susceptible, then they don't get the disease.
Bit of a tautology.
And I remember thinking at the time, well, that's a bit convenient, isn't it? It's like, well, why are you susceptible and not susceptible? So is this thing causing or is this the poor maggots on the dead body and it's just getting blamed for it, right? So, but because we can create evidence, we can create answers to say, well, no, we gave you this thing and you didn't get it. So it must have worked. Yeah. Right.
Well, it's just no different to necromancy. I mean,
yes.
I mean, it's religion, it's magic, it's science. The distinction gets very fine when you don't know what you're doing. I mean, you accumulate.
Yeah. Because not that you can actually look. No one's actually getting under. I mean, I don't know what people would think about this, but there's no one's getting under the microscope and seeing the lock and key happening and going, there we go, we worked it out, right? It's a guess of what we think the chemistry and the structure and the physicality of this is, and the way it's coming together, and we tell ourselves a story around what happened, and we say, therefore, that's the answer, etc. And it's only then when we get a weight of evidence to go, well, hang on a minute, we got another plague and Jupiter wasn't quite aligned, that we start to sort of go, well, maybe it's not working.
Yeah, I know. Well, I find it fascinating because with this spreading of oil on water, Benjamin Franklin was going backwards and forwards from England and France and England at the time of the Civil War, American Civil War, and he observed this problem of oil on water, throwing fat and old used stuff out the back of the ship, and calmed the waters. So surface tension was dropped by the fat from the oil from the kitchen. And he did that famous experiment when in England he went to this place called Clapham Pond, and he spread a drop of water, oil. So the oil spread on top of the pond, and he knew the area of the pond, so it spread out to form a single layer. So he deduced from the volume of the oil
of the drop
that would cover the area of the pond what the thickness of a molecule was.
Right. Yes.
And he was right. A very simple experiment. Okay, except that he really didn't do it. The story is told in the books, but in fact, he got bored and he went down to a house of ill repute with the ladies that he hung around with, and the experiment was done by an Englishman about 10 years later. Checked it, but his intuition was right.
So he came up with the idea.
Yeah, and did it.
And said, I did the experiment.
He said, oh, it'll work, yeah.
Right, okay.
That's confidence.
I've met some guys like that.
That's confidence.
Right. Okay.
But I mean, that whole
But
idea,
he was right, yes.
Yeah, interfacial tension and so on is as it gets into molecular forces, quantum mechanics and everything else. Although quantum mechanics is all stuffed up too. I can tell you about that later on.
We might, that's going to be a whole other session on its own.
I could just deal with quantum mechanics in half an hour.
Okay. Okay, so just sort of almost summarising to some degree, and then there's a couple other things I want to ask you about. But so my understanding of this now is that really the key here is if we can stop the virus coming in, or any viruses, but we'll use COVID as an example. If we can stop the virus coming in and out of the body, which is what looking at that sort of CO2 defense mechanism scenario, first of all, we're breathing out hot
No, it's
CO2.
coming out of the body. The virus is coming out.
Sure, but I mean, if just from a pre-infected scenario, we're all walking around with this CO2 armor. I'm imagining it because we're breathing out CO2. That's at 37 degrees, so that's bad for viruses trying to get in. We've got this endothelial surface layer and the lung surfactant layer stopping us. Okay, so that's our defense mechanism, and why we're not always totally infected. The virus is then coming in. It's knocking out this protein C, and on the way out,
it's picking it up.
Yeah, but it's knocking out the lungs, which is why the lungs aren't working.
That's right.
And then it's coated
in excess as it goes out from old people.
Yes,
there's heaps of virus because the virus isn't destroyed as it comes out.
Yes, it came in easily, got replicated plenty. Yeah. It's come out in high dose. Yeah. But when it's coming out, it's picking up this sort of protein C as well, so which means the next person that's getting it is dealing with a slightly different virus as well.
That's right.
Yep.
And so you don't get the same response so you'll go through a cycle, and then after the virus has been through a couple of cycles, different hosts,
it's kind of a different virus.
It's neutered, yeah. It
Yeah.
doesn't cause the same inflammatory response,
because it's already got this protein C, and
therefore
it's not going to pick up as much next time. Yeah.
Well, that's what we think. I mean,
yes.
Some of this is speculation. Some
of that might be Uranus coming in too.
That's right. Could be Uranus. Yeah, we don't know, but the story makes the COVID stuff isn't published yet. The stuff on endothelial surface layer is.
Yes.
But so it's not. We're speculating now, but it seems to make sense. All makes sense. It all fits together with the premature babies, with old people, with what the endothelial surface layer, the lung surfactant. All those things are missing from the thinking of the medicos.
Okay,
absolutely missing.
It is, and it goes back to what you said at the very beginning, this concept of cross-disciplinary scenario.
That's right.
Because there's almost this, at least from the outside, from what I see, there's almost this sort of idea of there's these like almost swimming lanes of science now, right? You've got your chemistry. So once upon a time, there's your chemistry and your physics and your biology, et cetera, and they've all sort of morphed into this huge complexity of specialization within, right? And so that, you're a nanochemist to some degree versus a astrophysicist versus a geneticist and so on and so on. And there's this kind of, and there's almost like, especially with the sort of war for ideas now that we have and the competition for resource to ideas, is there's this almost thing like saying, okay, well, look, if you're not an expert in genetics, so why would I listen to you when it comes to virus? Because we know viruses are an RNA, and that's a genetic thing, and therefore, if I want to understand how to combat this virus, I'm going to deal with virologists and people with training in genetics. And along comes the SOAP guy, right, the surface tension guy, who goes, well, if you just do this, it locks the front door, and they can't get in, and they can't get out.
That's right. Yeah.
And it's all done. But you, I'm not even sure what you are. You're obviously a colloid scientist, but you were a mathematician, physicist who's now dabbling in biology.
Yeah, I did work chemistry and biology. Yeah.
But you're approaching these disciplines, if you like, not as a chemist or a biologist or whatever. I'm a scientist, and I look for, in the same way Da Vinci or Newton or whatever it was, just I look at problems and I try and use what I understand about the way the world works to try and answer those problems.
You've got to be able to ask the right questions, and to do that, you've got to bring different disciplines to bear. And the science is terribly compartmentalised now. And I mean, even in genetics, you talk about that, and you talk about genes and so on. Now there's a new science called epigenetics.
Yes.
And so on.
Yep.
Well, what is that? It's saying DNA lives in a physicochemical environment that depends on salt and temperature and so on, and that changes its conformation, how it wraps up and ravels up, and it's just saying we need physical chemistry. It was missing. We needed Uranus, and so they're terribly protective of their own discipline too, and very dismissive of other people, especially medicos. They can't help it. They go like a rocket all the time, and their knowledge of, they don't know any science. They don't know the difference between a positive charge and a negative charge. They talk about sodium and potassium. They simply don't know. And there's all kinds of paradoxes that they deal with, but they don't know. But it's difficult to talk to them. It's like talking about technology transfer as a scientist. You talk to people that do technology transfer, they don't even know what a molecule is. So, but similarly with medicos. And,
we've talked about this a lot in terms of also not getting too caught up in the science. It's even a molecule, right? So the way I try and talk when I explain this to people is first thing you always have to keep in mind, and you really have to keep it in mind, is all these are made up concepts. There's no such thing as a molecule. That's just the story we tell ourselves around explaining it.
You can see molecules,
right?
you can measure them.
But in terms of that, but what we call a molecule is a bunch of elements sort of stuck together in a particular way. And we can understand, the charges, your positive, negative charges, and the effect of those things all sort of coming together. But that's just that's our explanation of it, right? So just because a positive and negative charge come together, or just because these molecules form in a different way, doesn't mean, a bit like you were talking about with the epigenetics, doesn't mean that in all circumstances that's going to play out exactly the same way, because that's true of the environment in which you're observing it.
Yeah, they change depending on the environment.
Yeah. And that's getting back to the water, the salt, the temperature sort of scenario.
Well, the molecular forces due to quantum mechanics, they're highly specific, and they're operating all the time. But it's a deep philosophical question you're touching on there, because it's a question of self and non-self. Where does one thing begin, another end? If you take molecules of water together with DNA or something, they're all connected together. They're all operating together. You can't talk about a DNA molecule without the water, which is hydrating it, and the hydration water changes it. It's part of the environment. You can't.
I guess that's kind of what I was in my.
Yeah, I know. I'm saying.
So we think of these things as specific
but
blocks.
They're not. They're all together.
Yes.
Okay. And there's another example I can tell you. I'll go back in this thing. Here's another thing as well. See this glycocalyx? It involves all these polymers. They're charged polymers. They interact, and these guys and the physical chemists don't know. We discovered some forces acting between long, thin polymers, charged polymers, called polyelectrolytes. They're highly long range, they're cooperative, and they work together, and they're highly conducting. They're operating in other things. People aren't aware of them, right? But they exist because they're derived from quantum mechanics, and we've seen them in action in different places. But people don't like Chinese medicine, right?
Well, I think can I just say, so I think the thing with the Chinese medicine scenarios, and again, relatively limited layman's thing. So Western medicine seems to me has been developed very much on the reductionist causality idea. Keep going down the rabbit hole until you find the causal mechanism. That is the problem, right? Whereas Chinese medicine is much more about everything's connected. Everything's a series of interconnected channels, flows, or whatever sort of thing. And so you can't mess with one thing without messing with everything else.
No, but this is not that. This is reductionism.
Yes.
You can introduce the Chinese medicine. These are highly conducting. They're lining all of these veins going all the way. They're connected right back.
But it's throughout your whole body though, right?
That's right. Okay, yes. So there's roots going around. Okay.
Yep.
They're highly conducting.
Yep.
There's electricity going up and down them, right? So if you put a pin in one place, you've got to stir up at another place. So there is this mechanism, connected, highly connected thing, which has different node points. So if there isn't Chinese medicine, you've got to ask why there isn't. Now, the medicos and the chemists and everyone say, this is rubbish, go back. But I say, look, if you've got your eyes open, you have to ask why there isn't Chinese medicine. There has to be. And if you look, it's connected to, fish that fire bullets and other ones, electric charges.
Yes.
They're on the outside of fish, okay? They have the same glycocalyx. It's It's an exothelial surface layer. But they have these things focused, and the fish can harvest them. Bang, they go straight to the source. So this electrical stuff is real, and it's reductionist, it's fine. When the medicos refuse to throw away reductionism, they're doing it because they're using a wrong paradigm. They're not using the correct physical chemistry. They're missing dissolved gas anyway. So that's why it doesn't work. But so my point is that I mean, things are staring you in the face all the time, and people don't recognise them because they're stuck in a particular mould. And my favourite example is a thing in France, the third greatest pilgrimage site through the medieval ages was a place called Rocamadour. It's on a beautiful river in the Loire Valley, and people used to go there to do pilgrimages, and you'd come to the bottom of this steep cliff, and there's these steps carved in the thing, so you go up there on your knees and up to the church and do your penance. And Henry II had to do it after he told his knights to kill Thomas à Becket, and so for his sins he had to go and do the penance and go the 1,000 steps up there. Well, if you go there on these steps, they're full of fossils, absolutely full of all manner of fossils. So people looking at these damn fossils for all this time and thought the earth was 4,004 years old, according to Bishop Ussher and the Bible, they're looking at these bloody things and they just think they're marvellous things. They didn't recognise them as fossils, living creatures, even though they could identify them with shells and so on. So if you've got a fixed mind,
you can walk past
anything. They were doing for centuries. They didn't recognise fossils. It had to wait until 1800 before people started to pick up on the idea of geology. And they found fossils of animals, so that made them, okay, it's real. It's not just things. And so we have this terrible problem with science, and it's reinforced by having to have grant applications, because you can't get any money unless you have a grant, and the grant has to be approved by all the other guys, the peer groups, who reinforce each other. So
but you also, in your grant, you also have to decide what it is you're going to get, what it is you're going to work on and discover, and you almost got to have an answer in mind, right?
You've got to have an answer in mind. Yeah. So you've got to do what everyone else does as well.
Isn't that almost the absolute opposite to the concept, though, right? Is that if you've already got an answer in mind, I mean, okay, maybe if you're Benjamin Franklin, you can, and sort of, but if you've already got an answer in mind, you know, so human psychology, or certainly behavioural psychology, would suggest to me that if you have an answer in mind, you're going to find that answer.
Yeah.
Right? Because of confirmation bias, availability bias, but all those things that make us want to remain consistent to what we already believe. The last thing you're going to do is...
It's against open inquiry. It's like religion.
Yes.
You have a dogma and you've got to reinforce it all the time.
Well, so there's two bits to that, right? There's you have to reinforce it if the incentives are there to do so, right? So it's a bit like, you know, things like the replication issue and those sorts of things that are now at least getting a little bit more sort of recognition in sort of science, to say, well, hang on a minute, if you just think about this from the point of view of saying, if you really are trying to find very unique and unusual and weird things, the chances of that actually being true have to be very remote. So if every time you do a study, or a lot of the time you're doing a study, you're finding these really weird and fanciful things, then that can't possibly be true. You can't have a high number of papers discovering something weird and new when that's all you're looking for. Most of the time, what you've got to be looking for, you've got to find a lot of things that just aren't true, right? If you're generally sort of going out there and looking with a proper open mind, many of the things that you think might be true, surely a reasonable portion of them have to not be true. Turns out, I thought this would be the case, and it's not the case. Whereas nearly all the time, or very, very high percentage of the time, people find the answers that they're looking for in the first place. Yeah. Surely statistically, that's the first hint, right? Is that if most of the time we find the answers we thought before we started, either what was the point in the first place, or maybe there's a bit of a bias in there where we're,
it's a terrible problem. A colleague of mine, Nobel Prize winner called Pierre de Gennes, he was in the Collège de France, very famous guy. And he was had very charismatic, had a great following. And for about 30 years, the French would always say, if they went to a seminar, they'd open a colloquium seminar. According to a theory of de Gennes, there is blah, 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 blah. We did this experiment and we confirmed the theory of de Gennes every time without fail. Regardless of the numbers, they change the numbers, to fit. But there's no question, always it was right. So you get this fixed track and people can't move. And they don't want to move too because it's hard work to move into a different field. I mean, this fellow I'm working with now, Brandon Reines, he's been struggling for years trying to find someone to talk to who understood anything. Well, he couldn't communicate with anyone that didn't understand that physical chemistry has actually changed and there's a new physical chemistry that we're using in water that works. But all the other stuff doesn't work. It's wrong. It's invalid because they got the same problem. And everybody's in it. There's all these tribes of the scientists. You can call it, they're all bunches of dopes, a bunch of chemists, a bunch of stupid medical guys, all kinds of dickheads. They're all bunches, the whole lot. And they're all in these different silos. And they do not communicate, and they don't want to communicate because it's hard work. And they don't understand, it's all been taken over by big machines. If it's not big machines, if you don't do neutron scattering, it's nothing. They don't have the idea that maybe there's some simple things like bubble-bubble interactions. If you open your eyes, they're everywhere that you can see. And the best science is first-order science, the simplest stuff. But to do that, you've got to be able to open your eyes and see it. So you've got to be trained to have an inquiring mind.
And so, does that mean then that is it as simple as saying, well, there's a bunch of, let's call them foundational sciences like, chemistry or physics or however you want to describe it, that anyone who wants to do what could be regarded as some form of science, be it medicine or, chemistry or astrophysics or whatever it is, that getting that grounding is that where we go astray? Is it the grounding is not there? We try and be too specialist too quickly?
I mean, medicos only get 1 year of a couple of lectures on physics and chemistry and so on, but they're dealing with physics and chemistry. But it's so they're pressed into specialist areas from the word go, so they can't do anything. The foundations are absolutely important. You need chemistry and physics to do biology, really. Oh, you can do biology, but forget.
Well, you don't know how. You don't know what it's a bit like. It'd be a bit like trying to be a builder and never having to, learn to measure and.
Yeah.
You don't know what nails and screws are.
That's right. Yeah.
Yeah.
That's correct. Yeah. Environmental scientists don't do any mathematics. They have a program that does it for them. They don't learn any mathematics. They don't learn any chemistry. They just have a computer program. They're working on the Barrier Reef. But it's more complicated than that, I think. Matt. I thought a lot about it. But when we talked about that, there's a huge problem with mathematics. Mathematics lost its way. It used to be, it called mathematics the queen of sciences. It was the centre of all physics and chemistry from Newton's time on. But they lost their way and they've gone off into pure pursuits that have got no connection with the physical world at all. In fact, they almost deliberately avoid the physical world. They'll make models for simple equations like epidemics and so on, but fundamental mathematics almost deliberately has no connection for philosophical reasons. They lost their way. So that's a big loss to all of science. And the other big loss to all of science is computers. And computers have been good and computers have been bad. I worked at IBM Research to finish my PhD, and I think they've been a terrible. They've been great for many things, for technology and so on, but they're a huge hindrance to thinking. Absolutely, it stops any thinking. People can't do any mathematical work anymore. They don't think. It's all on the computer. Biologists suffer from that terribly because they sell them computer programs to make models of proteins. And these models have got 100,000 molecular parameters in them, between a CH3 group, a methyl group, or between an amine group, a carboxyl group, molecular forces. So they run a computer program and they produce a pretty model in colour of a protein that they want for something and look for some active site. The models do not have any water in them. Well, the whole thing's critical. It depends on water. They don't have any salt in them.
So if the molecule, in this case, the molecule was, I don't know, what it's in a vacuum in space, I suppose, at this point in time.
That is.
That would be accurate.
That's what they think, yeah.
Right.
Yeah, and they've got these parameters. And if it doesn't work, well, they change the parameters. But that's got no predictability. And they sell these things.
But the problem with that, though, to a degree, right, is the unimaginable, well, first of all, the unimaginable complexity of that. So the downside to any computer program, right, is it's just a bunch of things talking to itself, right? So you have to decide what those components are in the first place and then the way that it'll talk to itself, right? So an algorithm is just a bunch of calculations based on how you decided to do those calculations in the first place. So,
but it's a stupid way of doing it. You can do it, you can make a theory.
Well, it's a very effective way of amplifying something very quickly, right?
No, but it's useless.
Sorry, it's useless in this particular context, right? There's a really good example I heard this many years ago in music. Is one of the way, and I use this to demonstrate because I think it's an effective way for people who aren't you to understand. So when music first started to become digital, so this sort of music compression in the MP3 codec, so the algorithm used to compress music down to smaller files so we could all run around with iPods, etc. And so this is wonderful. People got their music and whatever. And I remember in these early days of this compressed music, it was like, well, yeah, it's handy that I can have all my music with me, but it just sounded shit. It sounded terrible. And I'm like, I can't believe people think this is a good idea. And so I was sort of really perplexed with it. And then I heard a musician explain this, and he said, and he was talking about this because he was a let's call him a modern sort of rap sort of style musician. So they construct music. They don't put an orchestra together sort of thing necessarily, right? As they get on a computer and they build it. Anyway, so talking to this particular guy, and they're saying, oh, when you go on tour, what do you do, and how do you create your show? And he said, oh, well, we have to bring an orchestra. And they're like, well, what do you mean? That's not how you make the music, sort of thing. He says, yeah, but if you take music and all this complexity, and you compress it down into a single track, which is essentially what you want me to do on tour, and then you blow that back up again and play it through speakers in an auditorium, it sounds horrible because you've lost all of the nuances and all of the bits and pieces that made it alive. And so if I go on tour, I can't play a digital track to an auditorium, to a stadium. It just sounds horrible. I have to bring all of those individual sounds and play them all to the crowd. So just because it's digital music, don't think that I can. I remember watching it thinking, well, that explains why I'm listening to this MP3 and it's shit. And then as the algorithms got better and the codecs got better, essentially over time, then the compressed music kind of get better. But if you play compressed music, right, it is the same song. No doubt if you knew the song I played to you, that's exactly the song and I can hear all the components of it. But if you put that through an amplifier and a really good speaker system, and you do that with a compressed file versus even a more modern, less compressed file, and especially an uncompressed file, everybody can tell the difference. Everyone can say, that's not it. I remember having this discussion with a mate of mine. He bought this new car and he was really going on about the stereo and whatever. And we got in, he said, oh, listen to how great this stereo is. And plugged his iPhone in and he played it. And I was like, oh, hang on a minute. And he's like, what do you mean? It's awesome. I said, hang on a minute. So I had the same song on my phone, but I have an uncompressed version, right? So I said to him, let me play this song for you again. And I plugged it in and turned his on. And he's just like, holy shit. He suddenly heard just how rubbish his music was. Exactly the same song. And he's like, oh my God. And he's like, that's it. I have to go back and redo all of my music collection because I had no idea how bad that what I was listening to, and I was listening to it, when it better speakers and all sorts of stuff, but the source was so bad.
Yeah.
And I didn't realise that compression, someone told me that I could compress it down to this small thing or whatever, and then I got into this whole paradigm of, oh yeah, that's sounding good, and I got good headphones or speakers or whatever, and it's all sounding good. And it wasn't until someone came and showed me the difference of the algorithm that says there's 0.000% missing from this. But I take that full data and play it to you versus the 99.996% of data, and everybody can tell the difference. Everybody.
Oh, yeah.
And it's like, well, how good's your algorithm, right? So great if you want to have highly compressed and have it everywhere and you listen to crap speakers and whatever, that's not an issue. But to tell yourself that that's the same thing, just because it's insignificant statistically, the difference, yet everybody can hear the difference. To me, it's kind of like, well, therefore, whenever you don't have all of the information, you fundamentally don't have all the information. And if you're running a computer program, that's based on almost all the information, and you amplify that to your stadium, it's going to sound the same.
Yeah. Well, the computer programs in biology don't have any information. they're just curve fitting, because they've got nothing to do with anything. But they sell them, and they for millions of dollars. Generally Chinese programs, and they fool themselves. They got a pretty picture out of it, but it's got nothing to do with anything. They can't predict anything. It's awful stuff. And they can't ever do it because it's impossible to do. But there are other ways of doing the theory that includes all the electromagnetic forces and uses experimental data to do it. So you can actually measure the forces and see how they get together, see how they turn and lock together. But the way they do it with individual molecules is stupid. It's like saying, okay, I want to...
In isolation. It's the spherical cow in a vacuum.
It's like saying, if I want to understand Matt Batty and how he interacts with Christopher, what I'll do is I'll make a model of Matt Batty. I'll start with all the molecules. There's 100 billion molecules, and I'll run them on a computer, and I'll generate a Matt Batty, and at the same time I'll generate a Christopher, and I'll see how they operate. However, I'll get the wrong interactions, so this Matt Batty will be a weird kind of bastard for sure.
A different kind
So
of
I try to understand this interaction, and they're going at each other, trying to kill each other off because they're crazy. They're a wrong Matt Batty and a wrong Christopher.
So, but part isn't part of the we sort of got to this idea a while ago that everything's made of something, right? And so we went down this sort of idea of, okay, well, we're made of molecules, and molecules are you're made of atoms, and atoms are made of neutrons, electrons, and protons, and they're made of some sort of dense energy or forces, fundamental forces, sort of thing. And we keep going down this sort of theory of the only reason we didn't find that building block is because our microscope wasn't good enough. And if we now we found the next one, and I have at least the impression from say modern physics now is that, well, we stopped kind of looking for that, because every time we look for it, it's not really there. What tends to be there is some form of sort of dense energy or some sort of and even if you go back to looking at things like Heisenberg's uncertainty principle, right? To say, well, as soon as we measure the electron, we know where it is now, but we don't know anything about where it's going. And if you look at that from a law of entropy perspective, is like, well, if you really did know where everything was and where everything was going, well, then you really could project the whole universe and know where we'd be 10 years' time or 15 years' time or whatever. Mm. And so, because we can measure that doesn't happen.
I think quantum mechanics has got itself into a terrible mess. There's a lot of paradoxes there that are just plain silly. And I could go off about that, but it's got a lot of problems with it, quantum mechanics, and they're showing up all over the place. They make a big fuss and song and dance about Schrödinger's cat and all kinds of crap like that, but it's crap. It's rubbish, and they're basing the problem is that they're basing their theories, basing the work they're doing in quantum computing on something called Bell's theorem, which is like Schrödinger's cat, but it's complete rubbish, because the theory is at zero temperature, and nothing happens at zero temperature, by the way.
Absence of energy.
Absolute zero. So it's all based on zero temperature. As soon as you've got temperature in there, the world is completely different, and it makes sense. You're not into this crazy world. So the quantum computing is rubbish. Particle physics is rubbish, complete rubbish. It's based on a myth. You talk about I'd rather the people at Paris, really. But their theories are much better. Well, these guys are so bad with their theory, which is all the rage stuff. Well, that's my point in talking about this stuff. It's all the rage. It's all the popular stuff. Oh, quantum computing. Ah, Bell's theorem. Oh, big stuff. Yeah. Aren't we clever? Yeah, we're doing all this stuff. We got these holes in molecules, coherence, bullshit. But it's total rubbish, Matt. It really is, and they're blinded by their own bullshit. It doesn't. I mean, it if you've got any common sense, it doesn't make any sense. And you know what? It doesn't make any sense. It's rubbish.
So the idea of well, let's say something like spin, right? So the quantum entanglement sort of scenario, right?
That's Bell's theorem.
So the idea that, well, everything's got a balance, right? Have we at least got that bit right? That the universe has got a balance. You know,
what's the balance?
Well, in terms of, like, for example, for every it just takes spin as an example, right? So we get this entanglement sort of idea of saying, if we've got a particular, a photon or electron, or we've got something and we understand its spin, right? And that's all probabilistic at any point in time until we measure it. And so we measure the spin, we know that spin's up. Automatically we know that it's match pair, the spin is down. And, what Einstein called that sort of spooky action as a distance sort of scenario, right? So that we've got this scenario where we don't know the answers to things, but because everything has to balance, for every positive there's a negative sort of scenario, then once we have one measure, then we know the answer to the second?
Well, not quite, because as soon as you turn on a temperature, then the rest of the world comes in and says, I'm having a piece of the action too. And I've got a whole bunch of spins. I've got billions of spins, and they're all interacting with your spins. So your spins are not left alone. They're going to be knocked around by temperature. So it's damped immediately and disappeared. And all of I've done a lot of work on this kind of stuff in molecular forces, and the zero temperature stuff, the people use, the electrodynamics, it's all wrong. At finite temperature, it becomes completely different. It's like saying you forgot about dissolved gas. It's completely different. So the quantum mechanics have got the same problem.
Yeah, okay. So is this a little bit like what happens, with if we look at Einstein's general relativity, right? So as we get closer to the speed of light, the math starts to matter enough now that we start to, we don't notice it. We don't notice, time dilation or whatever at a sort of normal scale. But when we get close to the speed of light, we start to see it. Is that a similar dynamic when it comes to to temperature, to absolute zero? Is like we get down to that zero and we ignore it, and there we can see it in isolation. But if we add temperature to the equation, is there an effect there? Is that something we can?
I don't know. Probably. For example, I know that if you have a Galilean transformation, you go from one energy frame to another at a certain momentum. So you'd have if you're doing statistical mechanics, you have making what's called a partition function to construct the thermodynamics from the statistical mechanics of molecules. You have what's called a partition function, and that involves an exponential of a sum over energy states. So as you go from one frame to another, you change the energy becomes energy minus momentum times the velocity. Well, it turns out that if you include temperature, if you're doing statistical mechanics, that Galilean transformation is relativistically invariant. It's a Galilean because it involves the temperature, and the temperature changes.
The temperature can't be constant the whole way through.
The temperature changes. It changes too. as soon as you have a thing has a velocity. So the two cancel out. So a Galilean transformation is in fact relativistically invariant for momentum. But if you start to include spin and angular momentum, you are in deep trouble. There is no such thing as an equilibrium. You can't do statistical mechanics at equilibrium. It's not possible. And that's the source of the problem with all the expanding universe.
Yes.
They're playing around with this Coriolis force and so on and coming up with this terrible contradictions that don't make any sense because the whole theoretical basis to it is wrong.
Okay, so
there's a lot thing.
Is it just too complicated?
No, there's a lot of things to do.
Or you've got to. Is it that we're doing experiments now too clean? Right, that we're ignoring all of those things that because we can't model them, we don't understand their interaction. It's a bit like when you first learn science in high school. It's like, okay, well, ignore friction and ignore dissolved gas, or ignore all these things that either make the formula too complicated, or we don't actually have a formula that works, so don't put it in there. And at the scale that we're measuring and observing it, we don't notice any difference. Therefore, you can ignore that scenario, and then we get built into this idea of all these things that don't matter. And we chase down rabbit holes of getting more precise of the things we do know, ignoring all along.
Yeah, ignore all
and then you go out to the real world and you scale up, and the engineer goes, "It doesn't work."
Yeah.
And you go, "No, it was perfect in the lab."
That's right. And the biologists say, "No, reductionism doesn't work." It does work. You just got the wrong bloody theory, you idiot. It's just and the arrogance of these people that think they've got the right theory and don't question it. That's a big problem. They've got limited brains because they're not prepared to let anything else in.
So how do you? We talked a little about this in the past as well about how do you reach a point where the way I try and describe it to people, and because I'm just guessing from observing from people, especially people like you, is like you get to this point where now. What is it? And you Dunning-Kruger effect, I think you're talking about as well. You get to this, the crest of Mount Stupid, right? You know everything. And then you start falling down this hole of the valley of despair, realising, you know twice as much as you knew before, and all that does is teach you that you basically don't know anything now, right? So now you don't know nothing. And then you sort of slowly start working your way back up to, I'm knowing something. When you reach that point of saying, okay, that awareness of saying, there's almost an infinite amount of things to know. Is it that despair that people are sort of riling against and saying, well, I'm not going to admit to everyone I don't know anything, therefore I'm going to become an expert in something that no one can really challenge because, I can go off on some very specific area of knowledge that no one really has got an opportunity to chase, as opposed to saying, no, I don't know anything, but I'll keep experimenting to progress the general knowledge, I guess what I'm trying to get to is was there a point in your career or a point in your life where you sort of said, okay, well, I'm never going to be worried about the things that I don't know?
No, I'm a scientist. I want to know things. I'm not going to worry about them. I'm going to find them out.
Well, people seem to be, it's almost like, what do you know? And it's really reinforced now, I think, with this sort of like expertise, right? So, we want to know something about something, we want to get an expert. So let's get an expert in. And there's a really good example we talked about the other night about this guy he's a philosopher, right? So he was involved in one of the US congressional sort of hearings, and they were talking. This was a climate change thing, and this particular guy, he's a philosopher, but he's done a lot of work in terms of climate change and fossil fuels and all that sort of stuff. And he's sitting there talking to this sort of senator, and she's sort of saying, so who are you? What are you? Why are you here? Sort of thing. And he said, she goes, you got no scientific background. He goes, no, I'm a philosopher. And she's like, okay, well, what do we need a philosopher for, sort of thing, And he said, and his response was, well, I'm here to help you learn to think better. And she was like, well, we need scientists. We don't need philosophers, sort of thing. I don't need to know how to think. And he said, with all due respect, you do, right? You've got a thinking problem, not a science problem, sort of thing. And I found that as a really, not just a funny YouTube clip, funny because it's true, but it was a good example of how you think about things is way more important than what you think about, I suppose. And I think people seem to be a lot of people seem to be a little bit self-centered, like, my value is what I know as opposed to what I can learn, maybe is a way to describe it. Whereas the impression I always kind of get from you is much more a case of, well, okay, I know a lot of things, but I'm not really interested in what I know. I'm interested in what I don't know.
Yeah.
And I only know things so that I can use them to try and find out more things that I know. Do you find that, is that a summary of the problem to some degree, is that people are kind of like, well, once I get to this point of knowledge, it's almost like, well, if I get enough money, then I'll be okay. It's like, well, I get enough knowledge, I'm smart enough, and I'm an expert in something now, I can kind of rely on that, as opposed to, you know, a true expert is kind of like, oh, you know, apparently the answer is this, and they kind of go, well, maybe, yeah, we don't really know, you know.
I don't know. Most people reach, it's like psychologists say that most people reach a mental age of 14, the emotional age of 14, they stop.
Yep.
It's very rare for someone to reach an emotional age of 16. And for someone to get to 21 is really exceptional. People stop, okay? Scientists stop too. They stop at a certain level. And either they've got too many kids, or they're too lazy, or they don't have security, or they're just not interested. They stop with what they know and they stop there. That's it. They're not interested in anything else. And after a while, they've invested so much energy in publishing the particular area, they certainly don't want to question it, because that would cause all their other work into question. Stupid.
There was a really good article, and I don't even know how I got onto this particular article many years ago. It was in the New Yorker, and it was by I can't remember the guy's name now, but he published this series of articles because he had found an effect. And the article itself it's called "The Truth Wears Off," was the article in New Yorker, and it was basically a bunch of research where this bloke was sort of saying, "Hey, I discovered this effect, and then the more of it I did, and I published it, and I got famous, and it was great, and whatever. But then the more I sort of checked, more research I did, the more I found that it was sort of went from my results were being very statistically relevant to not so much anymore, and eventually I got down to chance." And he got really disturbed by this, and even there's a quote in the article where he says one of his mentors said to him, "Well, your problem was you kept looking. You know, once you found an answer, you worried about it not being true anymore." And so he got very obsessed with this idea of sort of saying, "Okay, well, how much does the truth wear off, right? So how much do we rely on, you know, p-values or this statistical measures that have been in many respects, in many cases, arbitrarily kind of like picked as that's a threshold, that's enough. And if you meet that threshold, you're done," as opposed to saying, well, no, surely we want to know what the truth is. And it turns out that the law of large numbers, they're bigger. That's a much bigger law than we realise, you know, that we can very easily get what seem to be very unlikely statistical measures. But because they do show up, when we get them, we think we've discovered something only to find out later on that it just wasn't true. We were just slightly left of the curve. And yeah, that's very unlikely to happen. But because we do this so often, the unlikely happens often enough, especially if you don't really test it, that we start to find that many of the things we thought were true turn out not to be true. And so there's a whole range of areas of science, if you like, but especially the less objectively measurable areas of science in particular, that then means, well, if you spend your life work, as this bloke did in some degrees, coming up with a theory or coming up with the result, that then other people find and replicate, which happened, and it turns out to be wrong, like surely there's a price to pay for A, being wrong in the first place, and B, telling everybody that turns out you didn't know anything.
Yeah.
Except the thing, he didn't make it up, like he did the work and whatever. How do you deal with that as a scientist? Like, how do you deal with the fact that, you know, your life's work could turn out to be, you know, early on in your career, I suppose. It's not certainly when you've been around.
I worked for years in molecular forces. I had the world's leading lab, right? It measured molecular forces, theories, experiment. We're the world famous lab and so on. And then I discovered in 2000 the theory is all wrong. So I published it.
Right.
And I was delighted. And then I found out later on that it was even worse because we found all this stuff with bubbles and dissolved gas. So the whole theory has turned, we've gone all the way through, fooled ourselves with, and labs all around the world saying this theory works, it's fine, physical chemistry. They still do, they still use it. I've published a lot, slating and not slating, but showing how it's all wrong. And there's a whole new world out there that if you know the new world or accept the changes that were missing, then you got much more.
But is that an advantage, but you were part of the finding out that it was wrong? Do you think that makes a difference? Whereas if, let's say, you know, one of your colleagues in Russia or US or something, it was when you find out you're wrong about something, is there a part of you that sort of is like, do you feel the loss, you know, in the sense of...
No, I'm just excited.
You're like, you beauty, I'm glad
Great, I found something new, yeah. No, not at all. It all suddenly makes sense, great.
But do you get that's not only is it not common, it's increasingly less so?
I don't know. I don't know if it's increasingly less so or not. Look, like quantum mechanics that Planck found out just doing his measurements, and the results were inevitable. Bang, just found it out. Our bubble stuff is completely not understood still, but it's fundamental that it's involved in everything. But no, it's delighted to find something new. It's first order science. Most people do second order science and they get bored with it. They run the computer program and so on. And so of course you get bored with it. So you feel sorry for them. I've been very lucky that I found a lot of new things, and that's great. I've been very fortunate to do that.